Good afternoon, everyone, and thank you to the organizers for giving me the opportunity to talk today. Of course, I would have preferred to be there in person, but a virus decided otherwise in the last moment. Anyway, nonetheless, I'm happy to share with you some results of my postdoc project that I started a year and a half ago at EMBL in the lab of ASAMICME and in close collaboration with the lab of Alba Munoz and the lab of Guillaume Salbon. So, in my postdoc, I am interested in the emergence of shape, morphogenesis, across evolution. So during development, from initially simple, embryos undergo a succession of finely orchestrated transformations that lead to the emergence of diverse shapes. The emergence of shape diversity is a long-standing question and complex question. And even though today biologists have acquired a lot of genetic data about what defines the polarity of organisms, and even though they looked at specific organs, understanding the diversification of body plans at the organismal scale still remains a big challenge. We lack unifying frameworks to tackle this question of shape at the scale of a whole organism. And one reason for that is the complexity of the body plans. In particular, the body plans, the shapes you see here of these vertebrae is quite complex. So to understand what defines the shape of organisms, we need a simplified version of the body plan. So for that, I propose to go back 600 million years ago, as I am focusing on a very old phylum called Cnidaria. Sorry, give me 1 second. Okay. Yeah, I'm focusing on an old phylum called Cnidaria. This is a sister group of bilaterians. So what are cnidarians? They comprise a large variety of animals, like sea anemones, corals, and another group, a large group called medusozoans, which comprises in particular hydroids, like the famous Hydra, Hydractinia, Clitia, and in particular also some jellyfish, as you can see on the right, on the bottom right of your screen. So as you can see there is a rich diversity of shapes within cnidarians. And however, these shapes have a lot in common. They are acquired from just two basic body units throughout their life cycle, the polyp and the medusa forms. So yeah, they are actually very similar. One has the mouth and the tentacles facing up, the polyp, whereas the medusa has the mouth and the tentacle facing down. So in particular, a unifying feature of these cnidarians is the polyp form. I mean here that all the species you can see here, even the jellyfish, at some point in their life cycle, they will transition from egg to polyp. Just as an example, Clitia, you see here in the bottom, this little jellyfish here, is actually bud from a colony of polyp here. This is the arrow on the top. Yeah, this is asexual reproduction, budding. So how do we transition from an egg to a polyp? I will emphasize this in the species I work with every day. So Nematostella is the species we have in the lab of ASAMICME. So when the eggs are fertilized, they undergo cell divisions and diverse early developmental processes during which they keep mostly a spherical form. At one point, they will reach the blastula stage. So what we call a blastula is an epithelium surrounding a cavity. After that, the embryo will gastrulate. So in the case of Nematostella, gastrulation occurs like this. It's just an invagination of the tissue in the cavity from one pole, the future mouth. And so at the end of gastrulation, the embryo acquires, let's say, its definite body plan. So it has one axis of symmetry, the oval, so-called oval axis. It's symmetric around this axis, and it has just 2 epithelial layers, which we call ectoderm and endoderm. So this is a relatively simple organization of the body compared to us, bilaterians or any vertebrates. Then there is an intermediate stage, the so-called planula larva, which is a swimming form of the animal, as you can see here. And as you can see, these planula are elongated along the same axis of symmetry, the orolaboral axis. So to complete the story, after these larvae, this planula will settle and undergo metamorphosis into a polyp. So today I won't talk at all about this metamorphosis. I will talk what I'm interested in, and what I will discuss today is this transition from a spherical gastrula to an elongated planula. And as you can see, the shapes of these planulas vary a lot across the phylogeny of cnidarians. These planula can be more or less symmetric. They can be narrow in the arboreal side, like or wide, like in Aptasia. They can even more narrow in the arboreal side, like in Hydractinia. The fact that the shapes of these planulas vary so much, and that they are a swimming form of the animal, makes this cnidarian a great model system to question not only tissue deformations, but also the functions of these tissue deformations on the organismal scale and the behavior of the animal, as they are a swimming form of the animal. So for my project, I am using this natural variation, this diversity of larval shapes, and I carry out comparative approaches. Namely, I am looking for common points and differences in the elongation process between the species, and I am to use mathematical modeling to find the parameters responsible for these differences of elongations. So, of course, I would love to understand all this transition you can see here, but so far, I have been able to work on these 2 species, the sea anemones, Nematostella. So as I said, we have it in the lab. This is the species I work with every day. And Aptasia is kindly provided from a collaboration with the lab of Annika Goods, which is in Heidelberg, of organismal studies, downtown Heidelberg. So I have described the dynamics of shape changes from gastrula to planula in these 2 species. Here they are. So what can we say about these shapes? The first thing that strikes the eye, I think, as I said, is that one species, Nematostella, gets thinner in the bottom, in the arboreal side, whereas the other one gets wider. And another thing that like curvature profiles. So here it's what you can see is curvature profiles average per stage. What I realized is that the position of the minimal curvature on the lateral side, the lateral minimal curvature is actually quite constant from planula 1, and is positioned more towards the oral side in Aptasia compared to Nematostella. Now, as I said, I want to use modeling to understand where does this shape changes dynamics, these shape changing differences come from. So when I visited the lab of Guillaume Salbreux in the beginning of my postdoc, I had the chance to talk with every one of his team, and in particular, there was Diana. I could talk with Diana Koromskaya, who was actually designing a model that is perfectly suited for my project. So as I told you, the body plan of my animal is like this. It's symmetric around an axis, and it has 2 epithelial sheets. Well, the model of Diana, the model she designed, studies axisymmetric deformation of the sphere. So she studies shape changes adapted to the symmetry of my animals, of cnidarians. And also, this is a model that is designed for epithelial morphogenesis. So in particular, the tissue, the model tissue is elastic with respect to area changes, resists bending, and is free with respect to shear. This is, so to go a bit more in details. So yeah, I just go back to mention that they published a preprint version of the model a bit more than a month ago. If you want to take a closer look, because I will just do a brief introduction of the model today. So this is a continuum description of the epithelium. These characteristics of epithelia, elasticity, bending resistance, and fluidity with respect to shear are associated to some parameters. And these are the constitutive equations of the model. So there is a tension tensor and a bending moment tensor. So there are many parameters in this model, but what Diana and Guillaume are focused on in this work are the implications, the roles of the parameters in red here, which are the active contributions, namely the internal tensions and torques in the tissue, but also, excuse me again, I need to, yeah, I have a little issue, okay. So the tension and torques in the tissue, and also are included the nematic components, which stem from the in-plane orientations of cytoskeletal elements within the epithelium. So today, I will mainly focus on this parameter, zeta C, which is in the bending moment tensor. And so zeta C is the isotropic bending moment. So what is the framework? I mean, how did Diana and Guillaume study these deformations? They do like this. They put an active region in the bottom, which in this case has an active bending moment, an active torque, and a passive region on the top in the blue. And by putting different extents, different patternings between this blue and red region, active and passive regions, and different intensities of the active contributions, they did simulations and established these morphospaces. So as you can see, by varying the extent of the active region in red, so on this axis or the intensity, we have a lot of different shapes. When we go beyond this bifurcation curve here, we have also changes in topology. I wanted to mention that. So for instance, here in the square, we have a constricting neck, so the shape divides in two. Or when we are in this circle here, at this position of the morphospace, we have a curling of the active port in the bottom until the two poles intersect. So large isotropic active bending moments can lead to changes in surface topology. What I want to emphasize in this figure here is that the higher extent of this active region, this abroad active bending moments, can invert the width profile. Namely, we can go from a shape that is thin in the bottom side to a shape that is wider in the bottom side. And that is reminiscent of what I observed between my larval shapes, planular shapes. So I want to test this. I've been testing if these simulations were relevant in the biology of my animals. So how can we do that? There are several works that that show that using the phosphorylation of myosin is a good proxy for for tension, and in particular, the differences between the apical and basal phosphorylation of myosin could be a good proxy for active bending moments. So I'm just citing this work from the lab of Guillaume Charras. So they have an apparatus like this. So in red, it is a layer of cells and DCK cells, which sits on top of a collagen layer. And when the cells digest the collagen, as you can see, the tissue will bend. They showed, and just briefly, they showed that this bending, for this bending to occur, this curling is driven by apico-basal heterogeneities in myosin phosphorylation. So if they block by a ROCK inhibitor, something that is upstream of controls the phosphorylation of myosin, if they block these apico-basal differences, the bending does not occur. Actually, they also mentioned that with this apparatus, by measuring the force that is necessary to keep the layer straight, they can estimate the active bending moment. So I use the same method, namely it is just an immunochemistry of phosphomyosin to estimate the active bending moment. So this is an illustration in aptasia. You can see that phosphomyosin is expressed in both apical and basal layers. And in this case, the bending moment can be approximated just with this formula. This is a product of the thickness of the layer times the differences between apical and basal tensions. So what I did is I just carried out, here are just 3D views of the myosin immunochemistry, where you can see the basal side and the apical side. What I did is just I repeated the same kind of experiments with the same antibody against phosphomyosin across across the gastrula to planula transition in both my species. And I'm interested in these three parameters I put here: H, gamma A, and gamma B. So in this cross section, you can see first that the thickness of the layer evolves. The elongation of these animals is marked by a lateral thinning of the ectoderm. First, the profile is quite homogeneous, and after, the layer gets thinner in the lateral side, like here. What I want to say is that in aptasia, this thinning of the ectoderm is shifted already. It gets thinner, more towards the oral side compared to nematostele. Regarding the basal side, the intensity is actually quite homogeneous, quite constant across the whole axis. So I just realized yesterday I had an issue with my quantification. You will have to trust me on this one. But regarding the basal side, gamma B here, it's constant mostly across the whole axis. There is some small enrichments in the oral side, but very late. For the last parameter, gamma A, here I put now full projections. As you can see, it is not constant across the axis. It's not homogeneous. There is an enrichment in the arboreal side. This is more, you can see it's more intense in the bottom. It goes, the extent of this arboreal the extent of this arboreal region is larger in aptasia, and sometimes it goes very large. Sometimes I have some late granulas of aptasia whose phosphorylation of myosin goes very high and takes more than 3/4 of the animal. So here are a few quantifications. And here, at planula 2, you can see that aptasia in aptasia in orange, this arboreal region with more intense phosphorylation of myosin is more extended. So in conclusion, the torque, the active bending moment, is located. There is an active bending moment located arborally and in a wider region in Aptasia compared to Nematostella. However, we could not predict these shape changes from the gastrula stage. At gastrula, the thing is homogeneous, both for thickness and the apical intensity of myosin phosphorylation. So my message here is that I could not have predicted the resulting shape changes from the analysis of the initial stage, the spherical stage. So I wonder, my question now is, could we predict these shape changes, differences from the spherical stage? So what I did is that I looked at gene expression to see if it foreshadows anything. So a lot of work has been done about the genetics of this animal. I'm showing here a diagram from a very recent paper. We did a very huge work to describe the patterning on the oral axis of Nematostella at gastrula. And I selected 3 genes here, FOXA, WIN2, and 636. And I repeated these experiments, but I did it not only at gastrula, but across the whole dynamics and also in both species in Aptasia. So I've been very happy last year to carry out this new technique of in situ. So until recently, it was really hard to combine different in situs to see different genes expressed at the same time. But with this new technique called HCR, so hybridization chain reaction, we are able to co-stain, to stain several genes at the same time. So this is a repetition of what you saw before, the 3 genes together. Across the whole dynamics, you can see that they stay expressed in different domains. Also, but what I think is that Asia is more. So I show you quantifications because that's what's interesting to predict something from the spherical. Here's 3 genes in both species separately. And if I focus just on you can indeed these 2 domains already. Yeah. So this we can combine this gene expression and okay. Can you hear me okay now? Yes. I hope it will be good. Yes. Okay. So I resume. My message here was that we have genes that pattern the embryo into distinct domains in both species, but the red region in the bottom is more extended in Aptasia. And so, yeah, we can combine this in situ for gene expression with the immunochemistry for myosin to see that indeed the blue region, WIN2, is associated to low phosphorylation in the apical side. So the blue region foreshadows a low active bending moment, a low torque. Here I just show joint quantifications to confirm that. You see that when WIN2 is highly expressed, myosin is the phosphorylation of myosin is low, and vice versa. So to conclude, at gastrula, so from the eggs to gastrula, the animals, the embryos are mostly spherical. We have a gene, the patterning at gastrula differs between both species. The red region in the bottom is more extended in Aptasia. And this red region foreshadows an active bending moment. And so I hypothesize that this fits the simulation of Diana and Guillaume's model, and that could explain the fact that one is thinner in the arboreal side and the other thicker. I also mentioned that I emphasized the fact that the minimal curvature is positioned more already in aptasia, while in this simulation, the minimal curvature is mostly located at the junction between the active and the passive region. So that would fit too. So to go further, to test further this hypothesis, so what we try to do is to transform one animal into the other, basically, to transform the shape, through genetic manipulations, to make nematostellar larvae look like an aptasial larvae. And for that, we use what people hypothesized as a gene regulatory network. And by knocking down some genes, we can shift the boundaries. I show you just 1 or 2 examples. So this is a normal case, the control. This is how 636 is normally expressed in Nematostella at gastrula. Well, by knocking down a gene, SP69, we can move this boundary more up to go closer to aptasia. And they showed that we still don't have a wider arboreal side, but we mostly equalize the width profile. I will do much more of these, repeat and do much more of these experiments when I go back to Heidelberg. Because we know, for instance, if we knock down 2 genes at the same time, that we can move this boundary even more up. What I still need to do, and I didn't talk about it because of time, is that the model also comprises nematic parameters. So here I show you simulations when we just put a nematic tension in the red regions. And as you can see, we have, it corresponds to an elongation or a flattening of the embryo. So I need to describe more the nematics, which can arise from many components of the cytoskeleton. But I just mentioned quickly that at the first stages in both species, the shape of cells is mostly oriented orthogonal to the axis of symmetry. So that corresponds, that fits with an elongation of the embryo. So there is a lot of work to do, because also the microtubules can generate nematics. And as you can see when you look at them from the poles, we have different kind of orientation. So here we have an aster orientation from the arboreal side. I need to describe this more finely, because I realize that the late stages, it's very dynamic. I just show you quickly, I think I took already too much time. So simulations for the nematic bending moment this time, so which can arise, for instance, if we have different orientations between the apical and the basal layers. And I will just finish by saying that we aim to understand what is the link between the shape and the behavior of the animals. So in this case, the swimming behavior of the animals. So yeah, I want to thank all the members of my teams. So I work mostly in the lab with the lab of Alba Munoz. I mentioned Guillaume Salbro and Diana Koromskaya, who did the modeling. And I want to say a huge thanks also to the lab of Annika Gus and Sébastien Rupp, who hands me the aptasia embryos that I showed you today. And thank you for your attention. I hope my connection was not too bad and the cut in the middle.
Thank you for your talk. So I have a question. Actually, I find it very interesting that you somehow try to link also this wind signaling to mechanical changes, because this is what we predicted via modeling for Hydra symmetry breaking and pattern formation. But do you have any clue how wind is influencing this myosin on all this stuff? What is the connection?
Honestly, I understand very little so far. So I'm looking again at the diagram. Yes, here. Tac. Yeah, so it's known that winds travel from the oval side, which is the site of gastrulation, until the aboval side. And the last wind expressed is wind 2 here. I just observed that in the wind 2 domain, we have the ectoderm that gets thinner and the myosin that gets less phosphorylated in the apical side, but I have no idea so far about the mechanism of action of wind 2 compared to these other winds here. So I did not mention
Is wind
it.
2 specific for nematostella, wind 2? Because wind 3 is very typical for development, like also in Hydra.
Yes.
And wind 2, I have never heard of wind 2, so it's specific for nematostella, do you know? Okay.
Yeah, so it's very interesting, I think, to look at the evolution of these genes across different species. Indeed, Win3 is very important in Hydra and in other species. Also, I showed all the hydroids, all the medusas, the ones I showed in the bottom. Win3 is very important and is patterned. There are some works like this. But
because I can tell you my hypothesis. I think that wind influence mechanics through beta-catenin. So I think it's beta-catenin and not wind, and beta-catenin is downstream, yes, from wind. But okay, we can discuss it another opportunity, maybe in Heidelberg.
Yeah, I would love to. I don't know. So beta-catenin, I understand that it's upstream. And so, but yeah, the winds, different winds will have different implications about the phosphorylation of myosin and the active bending moments. I can also I showed you in these two species that there is a shift already in aptasia. Wind 2 stops, is more oral. And when I designed my probes for the Institute, I realized that wind A, you can see here in gray, is absent in aptasia. So that may explain an oral shift and all these oral shift I showed you today. I would be happy to discuss it further.
Okay, thank you very much, Richard. Is there any online question?
So there is a technical question here. If you consider the nematic order at the end, the symmetry will be broken. How realistic is the axisymmetric assumption?
You say the nematic order at the end. Can you?
If you consider the nematic order at the end, what does it refer to?
What do you mean at the end, Benoit? Excuse me.
It's a question on Zoom, so I cannot.
Okay, sorry.
At the end of the talk.
I think it's referring to the fact that at the end you were showing some rod-like cells, if I'm not wrong, Axel.
At the end, so yeah, I heard you badly. So I understand that the symmetry of this axis symmetry, the cylindrical symmetry, implies that we have topological defects at both poles, +1 topological defects, which can be mostly asters like this, or vertices, the vortex. I don't know what you mean by it would break.
I don't know if you hear me. Is it? I mean, if you assume this axial symmetry, then it's probably true. But wouldn't it be much more natural to have like +1/2 defects if you don't force this?
I will need to. That's a great question. So yeah, I honestly need to read more about the dynamics of topological defects, etc. But what I understand is that, and I think it's the only constraint that Diana put in the model, is +1 topological defects at both ends, which can be actually, here are the two extreme cases. So it can be a +1 like this, which can be an aster, or a vortex like this. But this is, so yeah, that's what I put going further, because I like a fine description of the nematics at the pole. It's actually quite challenging to orient the embryos at the poles at this stage because of gravity. You know, my embryos are between a slide and a cover slip, and when they are elongated, they will tend to fall. This is the kind of things I will do as soon as I get back to Heidelberg, to manage them to, I have a technique now to manage them to fit vertically like this, to image them at both poles.
Okay, thank you very much, Richard.
But I think the only assumption, the only constraint they put regarding nematics is +1 topological defects at the poles.
Okay, thanks a lot. I think we should move to the next talk in the interest of time. Thank you again for the very interesting talk.